Thank you, everyone. Welcome to our second installment for the BioNeex webinar series. This round developed in partnership with Defined Health. So to get things started, I'd just like to give a quick introduction. My name is Armand Avatisian. I'm a client solutions advisor at BioNeex. My background's in biology and biomedical engineering. I've been with the BioNeex team for about 6 months or so. BioNeex itself is an online peer-to-peer platform that facilitates licensing and collaborative partnerships in the biopharma industry. We're based in Berlin, but we really have a global presence since we have our advisors stationed in New York and also Copenhagen. So the platform itself really supports the business development efforts for development stage biopharma companies by connecting them directly with potential licensing and co-development partners around the world, really. So we really provide them with the increased visibility and efficiency in forming these collaborations. So the platform opens these opportunities for biopharmaceutical developers, which we call compound providers, to display their novel compounds on a global scale. And on the other hand, we have the compound seekers, who are these biopharma companies who are looking to expand their R&D pipeline by providing them direct access to such innovative compounds that are available for licensing. So using our platform, providers are able to list their compounds based on designated categories like indication, mechanism of action, stage of development, and so on, and they'll immediately appear on their pipeline. Next, they can upload their contact details, and most importantly, this non-confidential document that discusses their compound, really. And so as a compound seeker, they can search through the pipeline for whatever compounds they like based on their company's needs and categories. They can find these potential compounds and directly contact with the providers themselves. So with that introduction, we can move directly into the talk itself. So we have Dr. Jeff Bockman with us, who is an executive vice president at Defined Health. He has his PhD from UC Berkeley and later transitioned into Defined Health, where he leads the oncology practice and manages a plethora of projects across the spectrum from early stage biotechs to top 10 pharma. So throughout the talk, if you have any questions, I'd like to ask for you to open up the chat. I'll show it right here, down below. There's a chat screen underneath. You can just type in your name and your company, and you yourself can ask the question towards the end of the talk, or if you prefer, you could just type your question in the chat box and Dr. Bockman will get to it afterwards. So without further ado, Dr. Bockman, I'll let you take it away, maybe give a bit more about Defined Health, and then we can just jump right into the talk.
Thank you, Armin. Will do. I think you have to stop sharing your screen so I can share mine.
Go ahead.
If I can find that on the panel somewhere.
On the bottom share screen.
Not coming up now. Technology.
Yeah. Maybe.
No.
Okay. Are you on a Mac?
No, I mean, it was doing it before. When I click more, I just don't see the share anymore. You see, oh, there we go. Okay.
You got it.
Thanks, everyone, for your patience. All right. Hopefully, we have action. All right. So here we go, and thanks everyone for joining in today. So as you can tell from the title, this is going to be a kind of overview, some analytics, some details, some thoughts and insights, reflecting a lot amount of the work that we do in immuno-oncology. And it really is an embarrassment of riches. And I've used this analogy or metaphor of kind of comparing it to Herod's food court because, of course, you know, there are a huge number of very attractive edible treats or attractive baubles, if you want to change the metaphor. And the issue is really for all the stakeholders, the biotechs, the investors, the pharma, to try and figure out which of these are really, at the end of the day, the ones that are going to be most attractive, most delicious, I guess, to stick with that. So for those of you who don't know Defined Health, we're one of the leading business development consultancies. We're a very bottom-up focused entity. Literally just the past few days, we have brand transitioned to Cello Health BioConsulting. We were acquired about a year ago this past February by Cello Health in the UK. And so now we are part of a much broader team and are able to cover the full spectrum from the early stage biotech pre-proof of concept space that we tend to play in, all the way up through the later stage pre-launch, launch, AdCom, MedCom work, and other aspects that are kind of the sweet spot of Cello Health proper. As I said, our core work really covers opportunity assessment, search and evaluation, portfolio and platform strategy. Again, very knowledge-based consultancy, bottom up, with a perspective on understanding the scientific rationale, pathways, and therapeutic modalities, and helping pre-commercial biotech understand how best to prioritize targets, programs, indications, to ultimately help them understand the value based on target product profiles, and for appropriate situations to do the full valuations for those programs. On the flip side, for large pharma and other entities, we're mostly looking, at least in oncology, externally. What is the next wave of innovation? What are the next interesting pathways and therapeutic modalities? How do we evaluate and vet those? Where do they position and fit into our large pharma portfolio, whether we're a have, as one might call them, with a checkpoint, or a have not. And so with Define Health now integrated into Cello BioHealth Consulting and Cello Health proper, we can kind of cover this whole spectrum of drug development. So that's it for the commercial. So disclaimer, Cello is a public company, but this is as much to reflect that these are, our opinions based on data, and much of the data I will be showing will be illustrative. So don't get hung up on the particularities, because by necessity, I can only share things that have a little bit of time that's elapsed between our developing some of these analytics. So that disclaimer, of course, also applies to this, which is an important part of immunotherapy, which is the discrimination of the self, meaning the host, the patient, from the non-self, which can, of course, be infectious agents, but in this particular case would be the non-self aspects of the cancer, the tumor itself. And of course, the MSA winners here for, Dougherty and Zinkernagel for their description of MHC. And my disclaimer of self from non-self, since I often get mistaken, and since you're actually seeing this as a live video, you don't know, Steve Burrell, Pete Townsend, and particularly here in New York for Steven Spielberg. That's the real me. All right, so first of all, I want to start off by congratulating, lauding, recognizing the tremendous advances that have happened in the past 5 years or so in oncology, not just in immuno-oncology. It's really been a tremendous set of advancements. So whether it's the academics, the drug developers, the financiers, the regulatory agencies, et cetera, it's really been an amazing time trying to advance management of cancer across many different tumor types and many different mechanisms and modalities. Now, clearly today we'll mostly be talking about immunotherapy, but that's not to say that that is the be and end all, and of course it isn't. So I do want to recognize that, 1908, so 110 years ago, we have this publication here based on recognition of Dr. Coley's work from what is now Memorial Sloan Kettering. So recognition that this Coley's toxin seemed to be doing something in a number of patients. What was it? That was unclear at the time. How would you produce it and sell it? Not so easy. How do you standardize it? So in the intervening 110 years, mostly this has been ignored. Until really the kind of past 20 or 30 years, except for a small cadre of, as they were called, tumor immunologists who kept kind of plugging away at this concept. But for most of the kind of current history of biopharma, really meaning from the 90s up through now, most of immuno-oncology was defined by either a few approved cytokines and related IL-2, interferon, and mostly by the long history of failure of cancer vaccines in the space up until kind of 2011 with a number of key approvals, which we'll talk about. So it is important at the outset here to emphasize to what degree immunotherapy or immuno-oncology, as it's called now, is a very diverse space. And whatever kind of bucket one might be talking about, whether cancer vaccines, or even checkpoints, or talking about modalities. There is a tremendous range of different options and therefore opportunities, which ultimately, hopefully, will prove good for patients to have more and more of these different targets, cell types, modalities approved to give us many more tools to mix and match. And of course, part of that mixing and matching, sorry, go back here a second, is to make sure that in this great diversity of the immune system and its interaction with the host, that we can address not just T cells and not just CTLs, but other T cells, B cells, innate, adaptive immunity, all the range of different cell types and pathways, secreted factors, et cetera, that are going to be relevant here. So when thinking about immuno-oncology, of course, one thing that drives a lot of the interest of the industry in it, of course, is the sales in the space. So on the left, you can see here a rough cut, not so easy to do. So even that rough cut is very challenging, requires a lot of manual curation to really kind of separate out what we will call non-IO and IO. And I know that's a somewhat artificial distinction, and one might make the case at the end of the day that anything that has ever worked meaningfully, let alone curatively or analogous to curatively in patients, depends on the engagement of the immune system. But for the purposes of thinking about this in an easier way, we've created the more classic IO, as it's defined now, versus non-IO. And I would emphasize that non-IO would include antibodies as they've been historically developed, meaning Rituxan, Herceptin, et cetera, even though it's well known that many of these probably benefit from or even require their effector function and the engagement of the immune system. So you can see that the bulk of sales, of course, are still coming from non-IO agents, but the growth in immuno-oncology dwarfs that of the non-IO segment. And of course, much of that growth is coming from the checkpoint inhibitors. And although there are a lot of projections for the potential value both to patients and as reflected ultimately in revenue for other approaches, in particular, say, adoptive cell therapies like CAR Ts, right now, those sales are relatively small. So as we go forward in time and look at some projections of the actual checkpoints themselves, we can see that, over the past really year or two, the dominance of Keytruda has kind of come to the fore, and really the two main players, of course, being Merck and BMS now, with many other players now, most recently being Regeneron Sanofi with their checkpoint inhibitor. And these, as I said, are really driving the growth of this category at this point in time. So I used this slide a couple of years ago at a Cancer Progress, the conference we run in the spring in New York, opening talk that I gave, just to emphasize how, incremental really, not that incremental can have value, but how incremental much of the historic advances have been with previous types of agents, whether chemo agents or first generation of targeted agents, whether large molecules or small molecules. And this was a review publication by Pat LaRusso and others. Well, when you still sometimes get asked by people, why all the interest in IO, I keep hearing that it doesn't work on all patients. Well, yes, that's true. But one thing is very clear. Where it does work and in which patients it does work, it can have very significant benefits. So it has really changed the nature of managed outcomes and hope for patients with melanoma, whether Opdivo or Keytruda or many of the others. It certainly has changed selected subset of patients, such as this kind of first, monumental approval, if you will, in some ways, this tumor agnostic MSI-high approval of Pembro in not just in colorectal cancer, although that's the data here. Lung cancer, of course, where very much also kind of night and day for the appropriate patients, and also the recognition, which somewhat contrasts with much of the historic belief that both academics and industry have had about lung cancer. One, that it wouldn't be that immunoresponsive. Two, that chemo would be detrimental to some type of immunotherapy. Well, in fact, we have here a disproving of that old mantra or dogma, which I would emphasize, again, this learning on the job that we are doing, or as Ira Melman calls it, the grand human experiment. We've got to keep our eyes open and be learning not just from successes, but learning from the failures, so we understand how best to deploy these and other agents. So while the success has not been universal across all different tumor types, as the plot here on the left shows, and this isn't comprehensive, doesn't show all, particularly the coldest of the kind of tumor types like prostate, but it is reflective of a lot of analytics that went into this up until this was done in this past summer. Just kind of showing you the spread just for OS, but we've done it for PFS and overall response rate. And on the right, the New England Journal publication kind of correlating more or less responsiveness, meaning ORR, with the mutational burden or TMB, which more or less mimics kind of the plot on the left. The idea being that there are certainly tumors that are more responsive, where more patients benefit more, and there are tumor types that are less responsive, where fewer patients benefit, and often, those benefits are smaller, meaning, lower duration, and less durable remissions, etc. So as people often say, and those of you on this webinar who are entrenched in the space know, people divide it into hot and cold tumors. I prefer to use the hotter and colder concept rather than the very definitive statement of hot and cold. And a lot of where people tend to focus will be kind of here in this, what we call sweet spot or Goldilocks space of kind of the lukewarm tumors, for a variety of reasons that I will touch on later in this call. So the idea, of course, where everyone has been spending their time, and I'm going to presume many of those on the call, whether on the biotech side with novel targets or modalities, or on the larger pharma side who either have or do not have checkpoints, but are trying best to deploy and improve the outcomes for patients with whatever organic or partnered assets they're working with, that we are trying to turn these colder tumors hotter. And that's a function of a number of factors, including making sure there are cells there, if they are not there, into the tumor microenvironment, that they can function and be active, and that ultimately they can target something on those tumor cells for elimination, most specifically antigens of one sort or another, so that there can be a tumor cell killing. And so there are lots of different approaches and pathways and modalities, the original Shannon-Mellman cancer immunity cycle, and different ways to get at this, and the interface of many types of checkpoints and co-stims, et cetera, from Drew Pardol's publication. And now there are many, many more of these targets, even from when these publications came out just in the past, 5 or so years. So we know, everyone knows, it's by and large all about combinations. And so the updated Chen and Mellman cancer immunity cycle, which is kind of useful for kind of conceptually thinking about these things, more or less buckets this challenge into issues of the, let's say, the hotter tumor, so kind of the lower hanging fruit being the inflamed situation, and then of course the more problematic settings of immune desert and immune excluded, which is where the idea is of trying to improve the activity by, in general, adding something on to what is more or less, one could consider now, the foundational IO backbone of some type of checkpoint inhibitor, anti-PD-1, PD-L1. When you step back and look at the pipeline in light of the idea of needing these combinations, you get an interesting reflection of where the activity is going right now. And one can see, of course, that there is a large amount still of cancer vaccine activity here in the red-orange color. Some of that is heritage, things that have been chugging along for many years. Many of those are newer, meaning in the past 5+ years, different types of immunization modalities and to some degree targets, although many of the targets have been classically and historically known. And the more recent interest in the space being driven by the neoantigen concept. But there are lots of other modalities, going back to this idea expressed before, that this is a space that both benefits from and creates the challenges of having a multiplicity of different types of approaches, both targeting innate and adaptive, whether they're cytokines, co-stimulatory agonists, other novel checkpoint inhibitors, adaptive cell therapies, oncolytic viruses, bispecifics, whether they're T-cell redirecting or dual functionality, double checkpoint, checkpoint co-stim, double co-stim, etc. So a lot of fascinating work going on, and we'll touch on that a bit more. And just again, to go back to this idea, this somewhat artificial dichotomy of IO and non-IO, you can see here that the majority of the pipeline still, when broadly looking, is non-IO, meaning it's next generation TKIs, new types of stratified patient setting targets directed towards mutations like BRCA, etc. Next generation chemo agents still, lots of antibodies that are at least not technically in this separation of IO and non-IO, IO types of approaches, epigenetic agents, etc. So right now it's still only about a third of the pipeline that is formally IO as we've designated it. This from the recent publication in Nature Reviews from CRI, which of course owes its heritage to Dr. Coley via his daughter, really reflects some of the massive increases that there have been. You can see a lot of it is occurring around additional T-cell targets. A lot of it is occurring around adoptive cellular therapy, mostly CAR-Ts, because of some of the tremendous successes in settings like ALL or more recently diffuse large B-cell. And also some of the huge increases that's one seeing with kind of bispecifics, though that increase is as much a function of being kind of a smaller level of activity, at least clinically active. Much of it has been historically preclinical. Most of the clinical has been the T-cell or other cell redirecting, but a whole range of other bispecifics will be entering over the next year that are kind of more of the IO-IO types of combinations. Here's another kind of reflection of that pipeline activity courtesy of Forbion. And you can see kind of the range of targets, many of which we're quite familiar with now, whether the OX40s or ICOS or GITR, LAGs, TIM, etc. And here's another reflection here of these targets as distributed by whether they're small molecules or antibodies, etc. And one of the things that we all need to stop and recognize, as the title says here, is that despite all this activity, and I'll show you more a reflection of how active those combinations have been, by and large, they have not been able to demonstrate definitive, or more specifically, kind of sustain their evidence of early signals and translate that in later clinical development into significant and reproducible outcomes. And of course, some have been outright failures, although I would emphasize it may well be, and probably may not be, a function of these targets being bad, but more a function of how we're deploying them, how the patients are being selected, how the trials are being conducted. And we'll touch on a bit of that. Now, of course, a little bit of the kind of pharma Me Too elephant in the closet can't be ignored either, and that is that there's still a huge amount of activity around just classic checkpoint, meaning anti-PDX, PD-1, PD-L1, whether antibodies, bispecifics, other types of approaches. And there are a number of reasons for that. Of course, a lot of people chasing, the big markets, not uncommon in pharma. The other aspect, the more kind of glass half full, strategic interpretation would be the ability with these follow-on agents for other people to be able to control the destiny, particularly if they've got novel agents that they would want to combine with some type of a checkpoint, not necessarily be tied to the getting on the dance card of a Merck, a BMS, or any of the other current players with approved checkpoint agents. So to some degree, some people have raised the question, Bruce Booth at our Cancer Progress meeting 2 years ago, are we facing a situation where checkpoints may become commoditized? Right now, the short answer is no. There does seem to be differences in how they behave, whether that's a function of their inherent biologic activity or how they're being deployed in clinical trials. Clearly, the labels are different. So for the moment, it's not just, go and choose your favorite cereal or any cereal, but that's certainly something to think about down the road in the future. This slide really emphasizes this idea of these combination therapies, and again, this is more data from CRI, not from their recent publication, but from the one kind of around or following SITC last year, that was in Annals of Oncology, just showing on the left, just the sheer growth in the number of combination trials, which has lots of implications for everybody in the space in terms of how one thinks about navigating and strategizing about clinical development. Where are the white spaces? How do you get enrolled? How do you compete just for patients, let alone ultimately, if you make it through, for the market? And on the right, and this is certainly outdated now because I know that there are probably over 800 trials. That's probably single agent and combination with Pembro, but a huge amount of kind of life cycle management being deployed by all the players in the space. Always been classic development strategy in oncology, and here it's just being kicked up, as Emerald would say, a notch or two or three. This from our own data, and these are only industry sponsored, only programs that have been updated since 2015, only combinations, so not single agent. So any type of combination, whether it's a checkpoint or other IO agent, added on to a standard of care, whatever that might be, an antibody, a chemo agent, an MTK, et cetera, or IO combinations in 2s or 3s or whatever it might be. And you can see the majority are still, being driven by combinations with checkpoint inhibitors, again, underscoring their idea as being foundational. This from the brand new Cowan analysis, just because they do some nice representation of key milestones coming up, just looking at lung, and this non-small cell lung, and this just underscores again the degree of activity, competitive intensity that is going on with a number of these readouts in red that would be pending. So anyone thinking about trying to fit into non-small cell lung cancer, by and large, needs to be thinking about not just how do I think of going into a later line, presumably in progressors or primary resistance to checkpoint inhibitors, but also needing to take into account all the other agents that are using that as either their proof of concept or ultimately, the clinically relevant space in which they're going to be deployed. So this competitive intensity is underscored here in these heat maps. Again, take these as simply being illustrative to indicate how much activity there is across the major tumor types by kind of broad therapeutic modalities, whether a bispecific, cancer vaccine, specific types of approaches like checkpoints, et cetera. And you can see that it is quite intense. Much of it still mirroring as biopharma development has historically, by and large, the larger tumor types having the more activity, but increasingly development across smaller segments and high unmet need segments, somewhat independent of their size, because everyone knows that oncology leads in terms of niche and orphan and suborphan types of development opportunities, whether that's through a stratified way or simply clinically defined by lines of therapy and patients that progress through their various levels of standard of care. So intensely competitive, that was solid. This is heme. And you can see that, again, this underscores on the left, much of the activity is around pretty well-known targets for, say, the antibody approaches. And on the right, many well-known, but not all of them well-established targets, say, for bispecifics or for adoptive cell therapies with, of course, CD19, CD22 leading the way, but other targets kind of following suit that people are attempting to pursue. Now, we can't ignore the fact that with all these combinations and with all the usual caveats about animal models in cancer and the extra emphasis of animal models, whether they're syngeneic or not, when looking at the immune system, because still most of these models are not dealing with human or human equivalent immune systems of mice. So we're often rushing forward into development, into clinical studies with even more imperfect knowledge than we've had before. And that means that there are failures and the need for kind of recalibration and maybe some reorientation of the hyped expectations that we've had. And, you know, I'm not going to spend a lot of time on these. We all know about the IDOs, but as I said before, that doesn't mean that that is a failed target. And I think it's important that we continue to develop and study these. And similarly, as reported by on the Evaluate Pharma Vantage blog from the recent SITC, where I was at of the past few days, some ongoing concerns about how valuable or not the Nectar data will be, given that it is a lower response rates that are being seen than had been seen in the earlier readouts, earlier studies. So there have been stumbles, maybe some outright failures, more a time, as I said, to recalibrate and think about what one needs to do and how best to deploy it. And I think that's important to underscore here. So first of all, you know, it's important to think of where the outright successes have been, and a lot of them have been recently, mostly at ESMO, with the TLR agonists. In fact, all the companies presented reasonable data, with Dynavax here, but also Idera, Checkmate, et cetera. So this is interesting, not only because it is kind of a well-known pathway, the DAMPs, the PAMPs, et cetera, the danger signals, if you will, but also the fact that these are given intratumorally, which is something that historically had not been high on the list of biopharma, save, of course, for the approval of Imlygic. And then so when we look at the big successes, the clear successes so far, other than, say, the approval of Yervoy, of IPI, with a checkpoint inhibitor, Opdivo, and use with other anti-PDX agents, it's been toll agents, and it's been chemo, as in the lung story, and it's been MTKIs or other types of kinases in combination with checkpoint inhibitors that have really kind of shown the benefit. And by and large, many of the other IO-IO combinations have not. So why? Could be the targets are bad. More likely, we haven't really deployed enough learning in the limited ability we can from the models. That's not, and there are a lot of learnings that can be from these preclinical models. So I use that word limited in quotes. And also thinking about what we can learn from the previous failures or stumbles or readings out of clinical studies. And one of those things clearly is that we probably have not thought well enough about combinations, sequencing, how to put these two agents together. And just an example here on the left, and that's also some of the work that Holbrook Court had shown in the past, was that sequence can matter. Giving things at the same time can be either non-valuable, if not detrimental. Changing the order also can abrogate activity and lead to, you know, either less activity or even in some cases, you know, adverse activity. And the same for thinking about modulating IDO, TDO, thinking about how best to do that, again, probably reflects thinking more in a more refined way about which patients to target and how. Now, all this activity in the pipeline, all this, all the clinical success, as well as some of the stumbles, is essentially feeding the need for and the continued need for deal making and newcos, et cetera, because we know it has to be combos, and we're still struggling to identify more and more combos. So not just with chemo, not just with MTKIs, not just TLR agents, but other combinations, whether it's oncolytic viruses, other checkpoints, co-stim agents, whatever it might be. So we are still seeing a huge amount of deal making in the IO space. So this is kind of the transition to a little bit of that talk for here, and then go through a few of these slides and then have time for some Q&A. So these are just kind of some qualitative examples of some recent both deals and then some outsized fundings that we're having in the space. Not even directly talking to right now about IPOs, but so, you know, very recently, of course, the Lilly and next year approach. So there are a lot of novel platforms that are coming forward of various sorts to try and both deploy new modalities against new targets, new modalities against older targets, better ways to exploit what we do know currently about the immune system, And also, even with the agents that right now may be being somewhat problematic, let's say, in their performance, but clearly demonstrating activity, some activity in non-responders or people who have progressed, ongoing deals such as the more recent Nectar tie-up now with Pfizer, subsequent to the well-known deal that they did last year with BMS. And of course, a lot of new cos continue to occur, and fundings for established companies, whether it's Compass Therapeutics representative here, a very large Series A in the summer to advance their unique platform looking at combinations, and they're moving their CD137 agonist program forward, or Allogene, of course, which everyone knows about in terms of taking out those assets from Pfizer that include the Selectus material, and so this reflects both biologics platforms, antibodies, cell therapies, cytokines, other approaches. So again, both representing the diversity and power that at least holds potential in this IO space. When we look at deals, and again, this is kind of a surrogate of interest and activity, what we saw in kind of the early days post the kind of the 2011 dawning of the new age of this immuno-oncology frenzy, I'll use that in quotes for the moment. So 2015, when you look at the top deals that had been done, and the ones in red, whether those are top deals by upfront or by kind of BioWorld dollars or total deal value, the vast majority were IO. When you look at 2016, again, vast majority IO for both. When you looked at 2017, we started to see what appeared to be a little bit of change in this. So while the majority of deals were still, in terms of total deal value, IO deals, the upfront had dropped to more like about 50%, which was suggestive at least of some maturing, maybe some backloading and a decreasing willingness to plop down a lot of money on very early, because these are often early deals in IO. They always have been in oncology, but increasingly very big and early deals in IO. And maybe it was a bit of a pullback from that. So whether that was some maturing of vision, or maybe it was some IO fatigue setting in. And so was very curious to see what the year-to-date 2018 numbers looked like. And when you look at them, well, in fact, it looks like the kind of the frenzy hasn't changed. And again, we're back to the vast majority of deals, whether the top deals defined by upfront value or total deal value are IO. So clearly, this hasn't slowed. And whether that kind of change from 2017 is kind of before all that deal activity was being driven by the potential and hype, before some of the reality started settling in about some of these combinations not working and underperforming. And now, of course, that they are, it raises the white space opportunity again, meaning we still need more targets. So we need to study the targets we know better, but certainly one option is to go after new targets and new modalities. And that may be what we're seeing here with this deal-making activity. And when you look at the number of deals, although the total number has gone down a bit, and this is 2017, 2016, and 2018 were only 3/4, a little over that of the way through the year. So we'll see how that pans out. But you can see how much is immuno-oncology versus non-IO, just in terms of number of deals. It was kind of even up more or less, but the value of the IO deals, even on smaller bases, is so significant that it's quite astounding. I pulled out our own analytics just to show some of this recent data that I saw Mark Edwards put out as being the guru of deal-making analytics. And you can see, again, reflecting by and large, but not solely across the phases here, how IO deals are either significantly or somewhat more richly valued than their non-IO counterparts, whether by looking at the average or the median. So still a lot of money going into IO. I don't have any data right now that I'm showing in terms of startups, but that's still very, very active. So we're coming off the heels of SITC. It was basically Friday through Sunday. And kind of looking at the early read in the wires would give someone a somewhat deflated view of, where the field is right now. And this is just, reflected here in, again, this Evaluate Pharma Vantage blog, talking about some of what had historically been very hot approaches, whether it's Sting, for example, or other approaches, and how, whether one believes the bubble has burst. I'm not sure the bubble has burst, but again, I think a certain maturing of vision and understanding of the challenges has happened. So as that balloon floats off, we can think about how, going back to my original Herod's food court and the food metaphor, this kind of bake-off has not yet yielded the masterpieces that many of us had hoped, and notwithstanding the combination successes with chemo or MTKIs, or now more recently the TLR agonist, there are a lot of agents that were much lauded, the co-stims, the checkpoints, the OX40s, GITRs, TIGITs, et cetera, that have yet to really demonstrate kind of or to live up to the expectations that were generated. So we have a situation where, we are having some stumbles and some wobbles and some things that are not coming out quite as beautiful and perfect as we would hope, at least not yet. And I'm sure that will change. And so much of the focus on other cell types and pathways outside of just CTL, CD8 cells, and other arms of the immune system, and particularly the innate, and certainly the focus on the tumor microenvironment and the idea of primary and acquired resistance is where much of this combination work continues to focus on, both with trying to better deploy the agents that have been studied and stumbled, as well as with brand new targets and modalities, with the hope that at some point we'll be able to get to many more substantive benefit for patients across more tumor types. So longer durable remissions, more patients having that, and hopefully more situations that are akin to a cure. And, we do have to be realistic that these changes, these significant paradigm shifts, do take a long time. If you just look at the history of development of monoclonal antibodies, it took quite some time for these to take off, many stumbles and missteps, a lot of kind of different perceptions as to what would really happen that turned out not to be true. Like, antibodies would just be kind of the passive targeting, and they would need a payload. And yet, the current leading antibodies are not like that. They're naked antibodies. So we're learning on the job always, because nature will always be smarter than we are. And so the idea that in the roughly 110 years from kind of when this field of thinking about the role of the immune system in cancer began to kind of the dawn of the new age, more or less in 2011, and thinking about, it took a century or so to get there, and, the rate of understanding, increase in development, discovery of new targets, et cetera. I think by 2021, it will be a very different world than even the one I'm talking about today. So with that, I want to thank all the great team at Define Health, the oncology team, the analysts, who of course aren't only for oncology, but probably spend perhaps more than they'd like in oncology, and the ones in gray are people who have moved on out of Define Health to other opportunities, but have still had significant contributions. So I want to thank everyone there. Of course, remind you of our role and how we might be able to help you and your companies going forward, and let you know about some upcoming additional events, including, of course, our Cancer Progress event this May, coming May in New York City, and 2 webinars, one that my colleague James Lee will be leading on Chinese biotech investing, and one that was just announced that I'll be leading with my colleague Sarome Lee. She and I will be co-moderating one on cancer vaccines on December 10th. So again, I thank everyone for your time, and I'll see if there are any questions that I can address in the time remaining. Yes, you can get access to the slides for sure. I'm reading backwards.
Yeah, we'll be following up afterwards. We'll send you guys all a recording of the talk so you can rewatch it or share it with your peers. So at this moment, if anyone has any questions, we could start with that. We have one It says, what is your perspective on subsegmenting populations using molecular diagnostics? And a sort of follow-up question on top of that, where do you see the role of companion diagnostics in general in immuno-oncology coming into play?
Yeah, and that's great. I'm glad someone asked that because clearly I only kind of indirectly referred to it in the course of this past 45 minutes or so. It is very clear that, well, first of all, let's just back up a second. Let's talk about kind of non-IO as it's classically defined, and clearly even there, despite the successes of the ALK inhibitors or EGFR mutant or I mentioned TRAC inhibitors, et cetera, and even with increasing testing and full genomic analysis, whether done by foundation or done at the institution, we're probably not deploying and understanding how best to treat patients with the non-IO agents we have. When we get to IO, we're still really in our infancy, trying to get beyond PDL expression, trying to understand better about whether it's MSI high or mutational burden, whether it's TMB or some other measure, whether it's inflamed signatures like Merck had or Merck in collaboration with NanoString, whether it's what Defiance is doing and Jerome Galan. We clearly will benefit significantly if we have a way to better understand which pathways in a given patient in the immune system and that immune system's interaction with the tumor are most in need of recalibrating, shall you say. And I think that in order to also do that, it benefits us, as I alluded to earlier, to have more tools approved and available. The more tools we have, the better we'll be able to perturb the system to the benefit of the patient once we find out how best to deploy those tools, whether used in combination, sequenced through lines of therapy, whatever that might be. So it is a big unmet need. Of course, the caveat always is that still to this day, not solely, but to a great degree, diagnostics remains undervalued, certainly compared to therapeutic development, which can be a challenge for those smaller companies trying to move forward in the space. So. Any other questions, Arma?
Yes, we have one from our BioNeex team who actually wants to ask a question. Would you please join in? Hello?
Yes.
Yes, so we have a question. You mentioned a lot of approvals need to be made in order to move forward. So the question asks, are there current regulatory pathways sort of adapted to these new approaches?
Is that a question? That's a question about therapeutic approaches, yes?
Yes, I believe so.
Well, yeah, I mean, I think, we must laud the agency, whether the FDA or otherwise, for a lot of significant changes that have happened over the past few years to really both accelerate the approvals, and that's not just the breakthrough designation, but also to really understand aspects of the value of bringing forward combinations, and not just one unproven combined with a known agent, but potentially two yet to be approved agents, knowing full well that it's not the case that, despite the fact that we certainly want to believe that agents are best moved forward if they have single agent activity, and that's been a bit of the discussion around IDO inhibitors, that there are certainly therapeutics that won't have much, if any, activity on their own, and yet do provide significant value when combined with other agents. So I think the agencies have been very open, or continue to modify their views to help this. Certainly a lot of things that they've done recently around cell and gene therapy have greatly facilitated those type of more complicated modalities. So, the short answer is that, I think, in the past, people have often used them as kind of a straw man or a punching bag to say, well, that's what's limiting development, et cetera, improving a challenge. But I think my opening slide of how many agents have been improved in the past 5 years, as well as data that we and others have generated showing you how many can get approval based on small, sometimes single-arm studies. So you can leap from a phase 1/2 to approval, or certainly from a proper controlled phase 2 into approval. So I think by and large it's a very good time. That's not to say there can't be improvements, but
right. You mentioned there was a slide earlier on talking about how with the use of, I think it was Opdivo, in certain patients didn't work, but in the patients that it did work, there was real, substantial improvements in response rates. So I suppose in the broadest sense possible, how do you think that we should approach those patients where checkpoint inhibitors like Opdivo and Pembro aren't effective on them? How can we target those patient groups?
Well, and that's, that's what, much of the activity in the pipeline right now is focusing on, is both what do we use to improve either the responsiveness of those patients who have, say, suboptimal responses. So either, they're not durable or only a small percentage are responding, and of that only a small percentage are durable. So almost all of the approaches that are being looked at are somehow looking either to kind of come in subsequent to use of a checkpoint, and whether those are progressors or whether that's primary resistance, to resensitize, and in most cases, either keep the patient on the checkpoint or add a checkpoint back on, or in other cases, if they have single agent activity, or to come in with other kind of novel combinations without the checkpoint. But that kind of post-treatment experience, post-checkpoint experienced approach is where a lot of the activity is. Some is also going on kind of frontline in combination with these checkpoints. But that's right now, that's where we're at. This goes back to that question about the stratification, whether we can learn better, who's going to be more responsive, and therefore make better decisions both for managing current patients, but as well as thinking about clinical trials based on getting beyond just PDL expression into other types of, whether it's mutational burden or, again, inflamed signature or other things. I mean, that's currently kind of the intersection. Right now, we're just doing this on the kind of a little bit empirically, and a little bit, it's a little bit of a black box, right? We've got novel targets, novel modalities, preclinical data suggests some type of activity. A biotech, of course, will be best if it can show activity in a checkpoint resistant type of model, and either resensitize or come in with single agent and show some activity there, hopefully across multiple models. That's what they need to be doing. That's a lot of what we're advising our, biotech clients on. And then how to think about, well, it's one thing to do those studies for your proof of concept, but, at the end of the day, is that proof of concept going to be a place that is going to be clinically and then ultimately commercially relevant, whether or not that biotech is going to bring it forward themselves, or what we call kind of proof of relevance. So these are all the things that, we, our clients, and everyone else is kind of thinking about day in, day out with these combinations, is how do we, what do we deploy to improve the activity or to reactivate responsiveness to checkpoints? Hopefully that answers the question, or sort of.
Indeed. So if anyone has any questions, please go ahead, submit it into the chat.
I'm actually gonna, and of course, if anyone doesn't want to ask it live, they can follow up with either the BioNeex team, the hosts, or follow up with me separately.
Absolutely.
All right.
All right. So, well,
I want to thank everyone for your attendance, and I appreciate it, and happy to follow up, as I said, offline with anyone, either directly or via BioNeex.
Yeah, definitely. Big thank you again to Dr. Jeff Bockman, to Define Health, one of the leading business development, strategy, and advising firms out there. And for the BioNeex team, a global online platform for pharmaceutical collaborations and licensing. So we'll follow up with everyone. We'll send you guys the recording so you can rewatch or share with your peers. And if there's any questions, you have our contact information. You can just send us or respond to the email. So thank you again, everyone, for joining, and have a wonderful day.
Thank you. And those slides will be available as a PDF. So again, reach out to BioNeex or reach out to me.
Yep.
Thank you, everyone. Bye-bye.